Pardon? These?
Right next to it.
This?
No, you were touching it.
Where?
That's
Oh. Well, that should be inside. I thought these were something squamous epithelium on the outside.
That's this one.
Okay. Ah, all right. Well, it should be on the inside. So here's a histology. Liver is very distinctive. You should only be, all you should have to be able to see is this and know it's liver. Because these clear areas right here, the sinusoids. All right. Now, if we have a larger view, you can see that all the sinusoids are leading to the central vein. There's very little additional connective tissue around the central vein. Whereas if we look at a portal triad area, there's additional more connective tissue. So let me show you another. This is an excellent portal triad structure. So additional connective tissue, several vessels. Notice this reminds you of the macula densa. That's the bile duct with its round nuclei rather than flattened nuclei. Portal vein, branch, hepatic artery branch. There's probably lymphatics there too. It's hard to say which because there's no blood cells in any of them. But you can see the three structures: portal vein, hepatic artery with a thicker wall, a thicker tunica media for the lumen, and then the bile duct. So this is showing a electromicrograph of an hepatocyte. We'll look a little closer at the edge of it. So a lot of mitochondria. This is a hepatocyte. This is the simple squamous cell of our sinusoid way down here. These are those microrugae, microfolds, and this would be the space of Dissay. So we're moving glucose through this space into the liver cell, albumin back out. Now we're going to look at the other end of the cell. Junction, see the desmosomes? Except for right here, this is the bile canaliculus. All right, what I drew right here. Where there's a gap, we add up all those gaps, and we have a long, small channel. It's not lined by any other. There's no simple squamous epithelium here lining. It's the plasma membrane of the hepatocyte forming the wall of the canaliculus. This is cancer of the liver, but I want to get to the duct system here. Take a picture of this if you need. So the liver is constantly producing the bile. The bilirubin pigments and the bile salt dysfunction we'll talk about on Thursday. And it's draining out of the liver. We'll talk about the hormones and nerve control of that process. But as it drains out, remember I said the small intestine, the duodenum, is going to be the recipient of that bile. Well, we really don't need the bile salts unless we have a high fat-containing diet. All right, cheese, danishes, and pizza, and so on. And so the bile ends up getting stored in the gallbladder. The gallbladder has a very thin wall of smooth muscle. Some of you may have palpated that when we did our sheet plex. It's lined with simple columnar epithelium. It doesn't make the bile, it just stores it. And then as a result of hormonal or nerve stimuli, that smooth muscle can contract and move additional bile into the intestine if there's lipids and we need to keep the lipid droplets. Essentially the bile salts maintain emulsification. So let's look at the pattern of the external extrahepatic ducts that are formed as it leads down to the small intestine. You guys got the diagrams here that you need? So from the right lobe of the liver, we get the right hepatic duct. And all these other bile ducts are coming together to form these larger ones. And from the left lobe of the liver, we get the left hepatic duct. And those join together to form a common hepatic duct. You can see this on the inferior surface of the large model here. So right hepatic duct. Left hepatic duct. And common hepatic duct. Okay. Usually the flow is out away from the liver through these three ducts. You can see them here and here. Then we have cyst means bladder. So leading to the gallbladder, we have the cystic duct. And this is bidirectional flow, because it flows into the gallbladder and it flows out of the gallbladder. The cystic duct joins with the common hepatic duct, passes actually through the pancreas, as you can see there. And this is the common bile duct. The blue structure I'm drawing is the pancreas. It's tucked into the head of the duodenum. So the common bile duct is going to join with secretions from the pancreas. Now the pancreas also forms as an outpouching of the digestive tube. One grows out to the right, one grows out to the left. But we don't see that. We only see structures to the left. So what happens is the right outpouching folds over and fuses with the left. And that's why we see a pattern of our, main pancreatic duct comes along like here. This is an exocrine structure. And then it makes a dog leg and comes down and joins with the common bile duct. The rest of this here is very small, all right? Continues, it's not really shown on this diagram, continues across as a much smaller diameter and empties into the duodenum as well. It doesn't pick up bile. And this is known as the accessory pancreatic duct. Where the two chemical substances, the pancreatic duct is going to be carrying bicarbonate ions and digestive enzymes. So where the two structures, the two materials flow together is identified as an ampulla, which is a dilated region. So the older name is ampulla of Vater. If you don't like to spell long words, you can use that term, the ampulla of Vater. Or the hepatopancreatic ampulla. If you don't remember Vater, you can remember where it is. It empties into the small intestine at a small mound known as the major duodenal papilla. And that's this little bump right here. So what comes in is typically a mixture of the bile salts, so on. So the sphincter of Oddi is just that part of the intestinal wall that allows the material to come in. If that sphincter is closed, the bile is going to back up and be stored in the gallbladder. So on Thursday, we'll describe the hormones and that control this movement and the enzymes that are contained there. This is just a view of gallstones, which are not painful when they're in the gallbladder itself. It's when they're in the ducts and you get contraction that becomes painful. Anybody doing a case study on gallstones? Okay, we'll talk about it a little bit more when we do physiology. It used to be the 3 or 4 inch surgery to gain access to the gallbladder. Now it can be done with several. It looks a lot more complicated and painful here actually. But these are 1/2 inch to 1 inch long incisions that can be just stitched up, and a lot less recovery time than cutting through the muscle tissue. The gallbladder pain is often a referred pain as the peritoneum becomes inflamed from the gallbladder. Anybody know where that pain would be felt? The scapular region. So often the inflammation is from the diaphragm, so the inflamed peritoneum of the diaphragm can be referred there. So we're looking here at the exocrine cells. Remember, our islet cells are endocrine. They're not going to go into the duct tissue. They're going to go into the blood. And these would be the acinar cells. So this is what is similar. You need to be able to tell the difference between pancreas and parotid salivary glands. They're going to look very similar, except pancreas is going to have our islet cells. And we saw way back then at the beginning of the course. So we're seeing the islet cells labeled number 2. Alrighty, so today's a major anatomy day, model day. I have a doctor's appointment at 10:30, so your illustrious Jeff is going to be here to give you your quiz, and I wrote it. If you don't trust him, I didn't write it, but I put the questions together. So let's look at taking the quiz, last over 11:30. Let's look at taking the quiz. You let Jeff know between 11 and 11:15 when you're ready, okay? Unless you're ready in 10 minutes, then you can tell him that. But the quiz is going to cover structures and even a review of some of the blood vessels through the stomach, okay? Through the material that we covered last week. I did have some liver stuff in there, but we just got to that, so I moved that stuff out. And it will be just, don't forget your mesenteries, greater-omentums and lesser-omentums, okay? So mouth structures, teeth structures, salivary glands, components of the submucosa, mucosa are typical GPAT. So I think there's enough in that to stop at the pylorus and pyloric sphincter of the stomach, and there won't be small intestine, liver, or pancreas. Okay, he's going to cover you guys for that, and then the lecture portion, first portion for the afternoon class, because I don't know, I haven't seen these guys before. Nothing wrong. This is the only appointment I can get.
Can we defer the quiz to Thursday?
I thought about that this morning,
Because I thought it was
but
seriously Thursday.
I know, I didn't study either.
I thought it was seriously Thursday. That's what she
because I shortened it, let's go ahead and do this one now. I thought about it, I was just, to tell the truth, thought you guys would all leave if I did do the quiz. Not everybody. But I want you to get this done because we have a limited amount of time. We can do an extra quiz on Thursday that you could, kind of take the best of both if you wish. But I want to encourage you to stay and work on the model material. We have our histology emphasis quiz a week from today. Okay, so we'll do. I don't mind doing a quiz on Thursday, but let's do. You can take the best of either or.
know, right?